Welcome back, folks. This is a video lecture on sex determination. It's just the next part of Mendelian genetics. You should have learned by now how simple letters, which represent genes, can be inherited through a pattern using a Punnett square. If you're not familiar with that, maybe it's a good time to just go back and watch other video lectures. So moving on to today's agenda, we have a straightforward and simple agenda. First, we're going to do some review of what it means to be dominant and what it means to be recessive. Then we're going to look at some 4 basic patterns of inheritance based on whether the condition is caused by the uppercase letter or the lowercase letter, right? And whether it relies on what the topic of today's lecture is, whether that particular gene lies on the sex chromosomes. Bullet point number 3, we're going to look at how sex is determined in mammals, and those will include us. Then, bullet point number 4, we're going to look at alternative systems that exist in nature. So we're not the only way in which sex is determined. Other creatures can determine sex in a multitude of other ways, which we need to just go over briefly. And then finally, we're going to look at the biochemical nature of X chromosome inactivation. Here we'll talk about how in a female human being there's a possibility for her to be a mosaic when it comes to X chromosome genetics. But we'll get there eventually. So let's carry on by looking at the first of these agenda topics. So this is a review. It's going to last 3 slides. It's very important. If any of this doesn't make sense, it's best to pause the video and then refer to your previous notes. The first thing to understand, and make sure you understand this absolutely clearly, is that we've been using letters so far to represent individual genes, right? So if I use the letter C, I'm referring to a particular gene on a particular chromosome in that particular organism. Just for review purposes, we believe that humans have between 22,000 and 25,000 genes. Each gene would therefore be a letter in this particular scheme. Now, it's going to be very difficult, almost impossible for you and I to deal with all the genes at once. So we divide and conquer. We just look at 1, 2, maybe 3 genes at a time. And that's all we can do. Otherwise, we have to use computers beyond that. So whenever we say A, we're referring to a particular characteristic or character. In this case, the example would be hair color. So A would represent the hair color of that creature. B would be another gene somewhere else. It could be on the same chromosome, but probably on a different chromosome. And that could represent eye color. Likewise, C would represent skin color, and D could even influence the behavior of that organism. So genes don't have to control a physical feature. Genes can also control biochemistry, like the presence or absence of enzymes, and then go even deeper than that and control your behavior by maybe interacting with the biochemistry controlling your brain and your nervous system. So we are pretty complex, but we have to start somewhere, and we start at the base. So we keep things nice and simple, right? So letters represent individual genes. One last thing to talk about genes before we move on. Gene is the most fundamental unit of inheritance that we have. So you can't inherit half of a gene. The smallest thing you can inherit is a single gene. But normally you inherit the full packet of genes from your parents, right? You get 1 set from your father and you get 1 set from your mother. Does that mean that you get 22,000 genes from your dad, and then you get a duplicate set of genes from your mother, but they may not be identical. There may be slight differences in the 2 complements. So you can get brown hair from your father, you can get blonde hair from your mother, right? So talking about these alternative versions of the gene, those are called alleles. Or alleles, right? Pronounce it how you like. So far, we've learned that whenever we use the uppercase of that particular letter, that means we have one allele. And we can use a lowercase of the same letter to represent the alternative form of the same gene, right? So in this particular case, since we were referring to A's up here as being a hair color characteristic, then these particular alternative forms of A would represent something called the trait. So traits are part of a characteristic. In this case, the trait will be brown hair and blonde hair. But you see some mistake here, or is that a mistake? We have more than one hair color. So how do we represent that? Well, we can't at the moment. We haven't learned how to do that. So we're going to keep things nice and simple. We're just going to consider two alternative forms of the same gene for the time being and nothing further. In the next few chapters, we'll introduce you how to refer to multiple alternative forms of the same gene. But with this particular system, you can either use a capital A or a lowercase a. There's nothing else, right? So we keep things nice and simple. So like I just said, the convention is to use a uppercase for a dominant trait. That is a trait that will show itself in the heterozygous condition. And then to use a lowercase letter to represent the recessive form of that same gene, right? The recessive trait. So we do know that brown hair is normally dominant to blonde hair. So this could represent brown hair in this example, and this could represent blonde hair. So we'll come back to that in a second. Let's just move on to the next slide. So let's talk about the recessive trait. So the recessive trait is normally represented by the lowercase letter, as we said earlier, right? So in a diploid organism, as we are, we have two sets of chromosomes. One from your father and one from your mother, right? One set each. Just to remind you, using the pen here, that humans have inside normal diploid cells 46 chromosomes. And those 46 chromosomes can be divided into two sets. So it's like having two copies of the textbook, right? Each page represents a chromosome. So you got one copy of the text from your father, from your dad. That's called your one set. And then you got another copy from your mother. So this will represent half the number of pages. So this will be 23. So 23 chromosomes came from your father, and 23 chromosomes have come from your mother. Now these 23 chromosomes have to be now divided into two subtypes. And those subtypes are called autosomes and sex chromosomes. But we'll come back to that in a few moments. Let's just finish off our conversation to do with the recessive allele. Using the eraser to remove some of this so we can make some space for what I need to do. Let's go back and use a different pen and just tell you that in the diploid organism system, each one of us should have two copies of every gene. So you could have, under the A system, where A comes in up capital and lowercase, the maximum number of permutations is 3. So in the population of humans, some of us could carry two chromosomes, each of which carries big A, big A. Some of us could carry chromosomes that are big A, small a. And the remainder will carry chromosomes that are small a, small a. And we already learnt in the past that this is called the homozygous dominant condition. This is called the homozygous recessive condition. And then finally in the middle, we have a single word to describe this, which is heterozygous. Okay? Now, of these three possibilities in the population, if the disease or the condition is tied to the lowercase, then the only people that will show that particular phenotype would be the people that are homozygous recessive. Okay? So that's what happens with a recessive trait. So if you have sickle cell disease is caused by a lowercase a compared to the normal version, which is manifest by having a capital A, right? So as far as I know from my testing, my genotype is big A with respect to sickle cell disease. But I do have a colleague who has a heterozygous condition, and then in Houston I used to know another colleague who had sickle cell syndrome, and he was small a, right? So remember that. Because we're diploid, we can have 3 possibilities in the population, but any one of us can only be one of these. So if I'm this, I can't be the other 2. If you're this, you can't be the other 2. You can't change your chromosomes once you're born. Indeed, you can't change your chromosomes from the time that you're conceived, right? So let's move on to the alternative, the flip side of the same coin. So that's called a dominant trait. So this could be a condition that's caused by the capital allele, not the lowercase allele. So the same thing, because we're diploid, there's 3 possibilities in the population. I just put them in very quickly. So there's 1 possibility for the population. There's the second, just like on the previous slide. And here's your third. In this particular case, if the disease is associated with the capital letter, then these individuals will definitely have the disease. And at the same time, so will these people. Because all it takes is 1 copy of the capital allele, and you will manifest that disease, right? And then, of course, these people, they won't have the disease because they don't have any capital A's. So that's called a dominant trait, okay? And what can I think of that does this? I think Duchenne muscular dystrophy or something like that, but we'll come across those examples later on in this course. So you can see a clear pattern, right? Here, only the people that are homozygous recessive will manifest a disease if it's a recessive disease. And over here, only people that have at least 1 copy of A, big A, will manifest a disease. So these are important things. Now, on the next slide, I'm going to talk about 2 more possibilities. So we have A, where the gene lies on the normal chromosomes, not the sex chromosomes. Remember I was telling you earlier that we have 23 chromosomes times 2, 1 from each parent, right? 22 of those chromosomes are called autosomes. I just used this word up here, right? And the remaining 1 from each parent is called the sex chromosome. There you go. So if you look at the whole condition inside a diploid cell, where you multiply this together, we will have 46 chromosomes inside a diploid cell. That's what this equals to. Plus, we have 1 sex chromosome from each parent out of the 46. So we will have 1 plus 1, and that will be 2 sex chromosomes. So a typical human being, you and I, will have 2 sex chromosomes inside our diploid cells, and we'll have 44 autosomes. It doesn't take much mathematics to decide that if genes are distributed across these chromosomes, most of our genes are present on the autosomes. But there are a number of genes, a couple of thousand, that are present on the sex chromosomes. Unfortunately, the sex chromosomes themselves are not the same. We have 2 versions of the sex chromosomes. So the sex chromosomes themselves are broken up into the X chromosome, and also they're broken up into the Y chromosome. And the 2 are not similar at all. There's a few areas that are similar, but those are not coding for genes. Most of the genes on those 2 are completely different to each other, and they code for different biochemistry. So that's what we're going to learn today. But let's just finish off this, right? So you could have a condition that's autosomal dominant. That means it's inherited as a capital letter on the autosomes. And following that is what we did in the past. And we'll give you better examples of that as we compare this to sex chromosome inheritance, right? So we have autosomal dominant. That's following, for instance, a big A. as it goes from parents to children to grandchildren. Then there's a second pattern, again from a previous slide, that's going to be called autosomal, the same word as above, so I'll type it in just in case we need to know how to spell it. Autosome, right? And autosomal, you can put AL on the end, and recessive. And that would be following the condition on the autosomes as a lowercase letter. Now we're going to introduce two more. So if you're looking at genes that are residing on these chromosomes, they too can be dominant and recessive, but they are called now sex-linked, right? Sex-linked. That means the gene is linked to the sex chromosome. And if that gene, whoops, if I can spell linked right, if that gene is a capital letter, like let's call it a B this time, big B, then it'll be sex-linked dominant, right? And if, the last category, if that particular characteristic or disease is sex-linked and it's going to be using the lowercase, then of course we call that sex-linked recessive. Why am I pointing this out to you? Because the patterns of inheritance can vary depending on which one of these four classes the particular condition or trait is associated with. And that's one of the skills that we want you to learn. Just by looking at a pattern to be able to decide which one of these it is. Or conversely, by being told which one of these it is to be able to deduce the pattern. That's one of the biggest skills in genetics. It's one of the top 5. So practice makes perfect, and that's what we'll be doing in this chapter and subsequent chapters. Okay. So if you're not sure what I've just said, I'll repeat it in the future. I'll do it in the next review, and then we'll show you some examples. So let's go on to look at sex determination in humans, mammals, right? So whatever happens in us happens in most mammals, not all mammals, there's a few exceptions. But most mammals, dogs, cats, etc., mice, rabbits, they will follow the same pattern. So what do we need to learn? Well, let me pull up something and we'll go from there. The best way to explain this, your chromosomal sex, is to give you a quick diagram. So let's draw a diagram. So on this side, we use the international symbol for a male. And then over here, we'll have the international symbol for a female. And what we're going to do, we're going to breed these two individuals together by sexual reproduction. And then we'll look at what happens before and when the children are born, right, or the offspring. So these are the parents. So in this particular case, we will assume that the father is XY and that the mother is XX. And right here you can see that this is the condition in mammals. So we call this the heterogametic sex, because as far as the sex chromosomes are concerned, males have the two alternative types. Hetero means different, just like big A, small a. So the males in our population, under normal circumstances, are heterogametic. And the females are homogametic, right? Like homozygous, homogametic. So when this male produces gametes, this X chromosome will end up in half his sperm. And the other chromosome, the Y chromosome, will end up through meiosis in the other half of his sperm in a ratio of 50-50. The female, on the other hand, only makes one type of egg, and that egg will only contain one of the two X chromosomes, again through meiosis. Now you can see what's going to happen next, right? When we draw a Punnett square, the Punnett square will have two columns because we have two types of sperm, but just one row because we only have one class of egg. So we put the mother's egg over here, and then we'll move this down here. So this is your X carrying sperm, and then you have your Y carrying sperm, right? And now you can see with respect to chromosomal sex, biological sex, the chromosomes will move down, and you'll have an XX here every time. So this will be a female, right? So let's use the international symbol there so we don't get confused. So all these babies will be females. And this particular column will always carry a Y, right, alongside an X, if everything goes normally. So all the boxes on this side will always be male. So the take-home message from this is that the father and his sperm, they determine the sex of the offspring. So this is pretty important in countries where boys are favored over girls still, unfortunately. The father normally blames the mother for producing a female offspring, but you can see now that the equation should be reversed. The father should blame himself because his sperm are responsible for generating the outcome of their child, right? So it's very important you do that. The other thing I would like you to practice as often as possible is to relay to yourself everything you've learned so far. So how many chromosomes are there going to be in the sperm? So the sperm will carry, well, you should know this by now, they will carry 23 chromosomes, each sperm, right? And how many chromosomes are there going to be in the egg? And the answer again is 23, okay? And those 23 are divided into two populations. 22 of those 23 chromosomes are going to be your autosomes. And one of them is going to be your sex chromosome in the egg. Likewise, in the sperm, we have 22 autosomes, and in each sperm, just one sex chromosome. You can see this one has an X, and that sperm over there will also have a sex chromosome, but in that case it's going to be a Y. So this is very important that you maintain a constant vigil over these numbers, because they just reinforce what you learned in the past, and they will then clarify things as you move forward. I can't emphasize that enough, that we must continue to do that as much as possible, right? So what else do we need to know? What happens when you inherit a Y chromosome? Well, the Y chromosome technically is much smaller than the X chromosome. So the Y chromosome has about 52 genes, last time I looked. In comparison, the X chromosome is much, much bigger, and it has about 1,500 genes along its length. So you can see the two chromosomes don't even carry the same number of genes, nor are they the same size, right? But as you learn later on, there are regions here that will match up with regions here. Just small areas of the two chromosomes, which are completely different, that they have large stretches of DNA that match each other. And those are going to be important in bringing these two chromosomes together during during meiosis and during the formation of gametes, right? But for now, most of the chromosomes are carrying information that is completely different to each other. So that's going to be important. So what happens? Well, sex is determined by the presence in mammals, by the presence of the Y chromosome. So if you have a Y chromosome, you're going to be exhibiting male-like features. If you don't have a Y chromosome, then you're going to be exhibiting female features. And the reason for that is staggering, right? Let me give you that. The reason for that is staggering because when we're first conceived, we are conceived to develop into a female, okay? So we're all going to develop into females unless you have a Y chromosome. Then about 7 weeks into your development, the gene, one of the genes, just one gene, a master gene on the Y chromosome turns itself on. And that gene has a name. It's called the sex determining region of the Y chromosome. And that's the name of the gene, SRY. So in females, you go through and you do not activate that gene and you end up becoming a female. In males, because of your Y chromosome, that gene will be activated and then you will be turned into a male. So the default pathway is a female. Unless you turn on the SRY gene, then you become a male. And we'll talk about complications of this pathway in another chapter, right? We have some athletes who are chromosomally female, but biologically male. How can that be? Or more importantly, the other way around. They're chromosomally male, but biologically female. So you can see something can go wrong in this pathway that can interfere with the final sex of this particular offspring, the baby. So this is a very important gene, right? So it's maybe the first gene that we really want you to know about in this course. So let's move down and get rid of some of this stuff. And what we're going to do now is we're going to compare how sex is determined in us with how sex is determined in other systems. So this is called the XY system, right? And here, the presence of the Y chromosome will make you into a male. Now you may be asking, what happens in other situations where chromosomes are inherited in different numbers? The answer is simple. If you have at least one Y chromosome, you will be a male. If you don't have a Y chromosome, you'll be a female, right? So let me give you a quick good example before I erase it. Let me change the pen. Suppose you were this, XXY. What particular sex would you be? Remember, the rule is simple. Do you have a Y inside your cells? And if you do, you'll be male-like, right? So this will be a male. How about this? And the 0 represents a missing chromosome. So when you see a 0 in a genotype, that means that chromosome cannot be found, it's missing. So in this case, that baby will only have one sex chromosome, and it's an X. Remember the rule, you must have a Y to become a female. So this individual will be a female by default, right? And one last one, just to match what you need to know. Suppose you were this. Yes, a triple X. We won't talk about the movie relationship, but triple X. So this individual, because they don't have a Y chromosome, would be female, okay? And having 3 copies of the X, well, we'll talk about that later today. So this is what happens with this system here, right? That's called the XY system, and mammals possess that. And there's other creatures on the planet that also have this system, and they can use that quite well, okay? So that's one way of doing it. So let's get rid of this down the bottom. Oops, get the right marker. So let's get rid of all this. So what else do we have? So we're going to do a comparison, right? We're going to contrast what we have with other creatures. So let's put a line to represent an alternative system. So let's call that system here the XO system. There you go. Maybe I should use a different pen. So that's called the XO system. And now you know what the O means. The O means that that chromosome is missing, right? Excuse me. So in this case, and the examples will be insects and stuff like that, a male would be basically an individual that's missing the second sex chromosome. So that will make it a male. So grasshoppers that are XO are male. So what would make it a female? Well, let's put it over here. So let's put males on this column, and then we'll put the female over here. So let's go back to the XY system. So to be a female, you will be XX. Okay? But the important thing is, in this case, that you have a Y chromosome. In the XO system, the important thing is that you don't have a second X chromosome. So having one X chromosome will make you male, but having 2 or more X chromosomes will make you female. And that's how simple that system is. Right? How does that work at the biochemical level? Well, what you're looking for is the relationship between the sex chromosomes and all the other chromosomes inside that cell. So we go back slightly here. We were talking about 22 autosomes and 1 sex chromosome, right? So there's a relationship. For every one of these, there's 22 of those. So that's called the sex chromosome to autosome ratio. So that's important down here too. In us, it's not important, right? It doesn't work that way. But in these XO creatures, these animals, it does work that way. So what you're looking at is how many X's do you have to your autosomes, right? So if you have only 1 X to your autosomes, you'll be a male. If you have 2 X's to your autosomes, you'll be a female. There you go. That's all it takes. So technically in the lab, under experimental conditions, I could take an egg that's got XX, remove one of the X, and then that creature, as it develops into an insect or something, will become a male because I've reduced the ratio of sex chromosomes to autosomes. Okay. What other system do we have? So let's bring in our pencil. And in this case, oops, I'm going to the wrong slide. So in this case, we're going to look at an alternative system, which is called the ZW system, right? The ZW. So here we go. So this operates mostly in birds, like chickens and parrots and stuff like that, right? In here, unfortunately for us, to be male, you have to have 2 copies of the Z chromosome. That'll make you a male. So can you guess what it takes to be a female under this system here? Yes, you need a copy of the other. So it appears that sometime in the history of the evolution of animals, one branch chose to use the XY system, and another branch of animals chose to use the ZW system. So over here, to be a female, you would be a ZW. That's all you need to know for now, right? So as we said to you earlier, we use those terms, heterogametic and homogametic. Well, males in this system are heterogametic, but in this system here, the males are homogametic. It's the female that's heterogametic because she has 2 different chromosome designations, right? So it's flipped. And like I said, next time you eat a KFC, think about that. So what else do we have? Well, we have more complicated systems. Let's quickly put them together since we have to move on, right? So in other creatures, we have something called the haploid system. Yes, that means one sex only contains 1 set of chromosomes, and the other sex contains 2 sets of chromosomes. Sets, right? So in this case, the males will be just N, 1 set of chromosomes. That'll make you male. And if you have the normal double set of chromosomes in a diploid condition, 2N, that'll make you female. Right? So what are we talking about here? Honeybees are the best example. So the drones, the unfertilized eggs that become the males, they are a single set of chromosomes. And I think in honeybees, N equals 16, right? So they have 16 chromosomes inside their cells. Females, on the other hand, they have twice as many. They have 32. So if you develop from a fertilized egg, you'll become a female. If you develop from an unfertilized egg in the queen bee, then you'll become a male. So that system also exists around us, just to complicate our lives, right? And as you know, bees are insects. So some insects do it this way, some insects do it that way. And it's just what it is out there in the world. But hang on a second. There's two other quick mechanisms that we've uncovered. So let's put them into, oh my God, it's getting a bit complex, right? So the next one, and we're going to leave space for another one down here. The next one is due to the environment. So in this case, it's going to be temperature. That's right, temperature dependent. So this normally applies to many reptiles and some fish, not all fish. And what we find here is that at one temperature, if the egg is incubated at one temperature, it'll become a male. If that same egg happens to be incubated at a different temperature, it'll become a female. And I think your book talks about alligator, the American alligator. That's a classic example, right? So if you incubate that at 33 degrees Celsius, and you keep it at 33 degrees Celsius from the time that the egg is laid until it hatches, it'll come out as a male. If you do it at higher temperature or lower temperature than that, by a few degrees, like 31 degrees or 35 or 36, then it'll become a female. Why? Well, that's the way that those particular species have evolved. And it allows those species to respond to different environmental conditions. So they're more responsive than we may be with the XY system. In the same vein, there's one last one that we want to consider, right? And that's the fourth, the sixth one, right? So let's pick the color. So that one there will be to do with behavior. Yes. And this is my favorite because it reminds me of the movie Nemo, right? So the behavior of the creature in its environment, in its colony, will decide whether it's going to be a male or female. So in this case, the adult is able to change sex simply because of its relationship with other members of the population. How does that work? Well, in this case, Nemo, the fish, the clownfish, is able to change from a male to a female if the female in its colony dies, right? So if you're a male and you're swimming around with a female and something happens to that female, then the males will become females and any babies they have will develop into males, adult males, right? So this way, you're not ever without a female. It is amazing, but it is what it is, right? So what you've seen here is an explanation of how sex is determined in the animal world under various conditions. Incidentally, what I haven't spoken about are non-animal creatures. So plants will be the other branch of multicellular creatures that we have around us. How about them? Well, most flowering plants, they have both types of flower on the same tree or the same bush. So they have both, right? Some plants, some trees are either male or female. So one tree will only produce the pollen, and another tree at a distant location or close by will have the stigma and style to receive that pollen. So in this case, each tree is acting like an animal, like you and me, right? So that's also present, but that's a rarity in the plant kingdom. So those trees are called dioecious, right? Dioecious, yeah. So that just means that they're two separate. But most trees, a single, like a pine tree, it's not a flowering tree, but it's a pine tree. It will be male pine cones and female pine cones, right? And they can cross-fertilize, self-fertilize in between the two types of cone. Okay, so remember that there are some other creatures on the planet too that perform sexual reproduction, so they don't all fall into this particular grouping. So let's take our understanding of chromosome structure, cytology. Cytology is the study of the structure of cells. Let's apply that and let's piggyback that with biochemistry, because we have to do that in science. We have to bring the different branches of science together so that we can explain as a single entity everything that's going on. So the last part, the final slide here, is going to look at some biochemistry, right? And in particular, we're going to concentrate on us, mammals, humans in particular, and we look at X chromosome inactivation, right? That's what this abbreviation stands for. So again, a graphic will help us here. So let's assume that we have a baby, and inside that baby, we have 22 normal autosomes, plus we have an X and a Y. So in this particular case, the ratio between the X chromosome and the other 22 is going to be 22 to 1, right? For every 22 of those chromosomes, we have one sex chromosome. So this person here will be a male, and everything will be fine. Suppose we have another baby over here. And in this case, that baby also has 22 pairs of normal autosomes. But this time, the baby is an XX. So you can clearly see that this baby over here will be a male, and this normal baby will be a female. So far everything is fine, just like we had way back here, right? So XY would make into a male, and XX would make you into a female. But how does that work at the biochemical level? This is what you need to know, right? In this case, the ratio between these two and these two, the X's to the other side, is now 11 to 1, right? So there's, for every 22 of these, there's 2 of these. So 22 to 2, but you can cut that down to 11 to 1, right? So this ratio in a female is completely different to the ratio in a male. And that is a problem. Our cells want a ratio of 22 to 1 in order to work normally. So males are fine. They pass the test from the time that they are conceived. Females fail the test. So they have to take some action during development to bring that ratio back to what it should be. And the default ratio is 22 to 1. So how do they do that? And the answer in humans is simple. Or is it? Well, it's straightforward. Either this X chromosome is turned off, or this one is turned off. You have to turn one of these two off. As soon as you turn one of these off, now your ratio goes back to this. When does this happen? I'm not sure. I think it happens in the seventh week of life. I can look it up for you. But it happens during development, when you're still inside the womb. By then, a large proportion, maybe the second trimester, a large proportion of the baby's body, the female baby's body, has begun to form, right? So you have lots of cells. Some of them, they will have this X chromosome turned off. In others, randomly, you'll have this one turned off. So in humans, it's a random process as to which one you turn off. So when this particular baby is born, if that baby happens to carry small a on this chromosome, say from the father, and she happens to have a big A allele on this chromosome, which she happened to get from her mother, then whichever X chromosome is turned off in that particular cell will then be inherited by all the other descendants of that cell. So you can imagine that you haven't started forming the arm of the baby yet, but you have a small bud in the embryo, and that's going to give rise to the arm. Maybe this part of the arm, it comes from one X being turned off, and this part of the arm comes from the other X being turned off. So these will have the big A active on the X chromosome, and these tissues over here will have the small a. So there's a possibility that a large number of females in the human population are what we call mosaics, right? Mosaics. That means they have tissue within their bodies that has different biochemistry. And that different biochemistry can cause sometimes different parts of the body to be composed of different chemistries. The classic example of this is the tortoiseshell cat, the calico cat, right? So the calico cat has some areas where one X chromosome is turned off, and that makes that part of the body black. And then it has other parts which have the other X chromosome turned off, and that causes that part of the body to be orange. So you can clearly see that in the case of the cat, but it's not so obvious when you look at females in the population. Now, that's only true if you carry different copies of the gene on your two different chromosomes, right? If you have big A here and big A here, it won't make any difference. But remember, there's 1,500 genes up and down the chromosome, and any one of them could be present in the heterozygous condition. Okay, so that's one way we do it, right? And this ratio is called dosage compensation. So the biochemistry has to compensate for the dose. If you have two chromosomes, two X chromosomes, both working, they're both going to be producing biochemistry, and that's going to reduce the ratio. But if you turn one off and shut it down by tightly packaging it and turning it into a solid structure, which can never be reactivated, whereas the other X chromosome is working away nicely inside the cell, then that's called dosage compensation. Just to show you this, on this last slide, I have introduced two images. The one on the right shows you a cell from this cat, or it could be from a human being, but this one comes from a cat. You can see the X chromosome that has been shut down, the technical name is inactivated, is present here as a highly dense chromatin, right? So it gets highly condensed and it forms this structure called a bar body. So let me show you how that works. So in a normal cat, that's female, you would shut down one of these two, right? Randomly, but every cell will have one of these two shut down. So this will lead to one bar body, right? Bar body. If a cat happens to be triple X, which can happen, then you'll have two of these X's shut down in that particular cell, randomly. So you'll have two bar bodies. And there are also cats, believe it or not, that can have up to 5 X chromosomes, like this. And then the question becomes, how many bar bodies will we find inside the cells of those cats? And the answer is 4. 4 bar bodies, right? I have a side question for you. If you have somebody who has this configuration, and by the way, that's known as Turner syndrome, after the first patient that had it, how many bar bodies would you find? Remember, the 0 stands for nothing. So there's no second chromosome, no second sex chromosome in there. And the answer is 0. Yeah, you won't have any bar bodies, and every cell of that particular female will be made from this X chromosome being active, right? So that's how we can explain bar bodies with respect to the inactivation of chromosomes. And here you can see a calico cat. So here you have the black version of the X chromosome active, and over here you have the orange version of the X chromosome orange active, right? So here the orange one has been shut down into a bar body, and in these skin cells, the black one has been shut down into a bar body. Lastly, this is not the only way that this can be completed. In marsupials, for instance, if the father donates this X chromosome and the mother donates this X chromosome in a mating, and the baby gets these 2 X chromosomes here, this is the mother's and this is the father's. In marsupials, this X chromosome always forms the bar body, the one that comes from the father. So the baby is entirely made up of any genes that are inherited from the mother's sex chromosome. So that's how it works in those creatures. But guess what? There's other alternatives too. And will you be tested on them? Not quite, but it's still nice to know that in other creatures, say the male is like this in these flies, and then the female is like this. In the flies, we've learned that the wild type, the normal condition is this one. So you have to have both your X chromosomes running in order to have a normal biochemistry, the normal dosage. So what about these male flies? How do they then compensate? Well, what they do is they hyperdrive the genes from this X chromosome. So this chromosome is turned up to be twice as active, right? So this is producing 1 unit of enzyme, this is producing 1 unit of enzyme. That keeps the female cells happy. Here, this one now is ramped up to produce twice as much from every single gene. So that way you get the level of expression. Expression means the activation of genes. The expression of the genes is times 2. And that way you can now match the male cells to the female cells. All right? And of course, there's other systems too, if you read your notes. The final thing I want to say, of course, is to talk about a gene that's present on the X chromosome that's going to be becoming a bar body, right? So let's go back to the human condition. So here's a human female cell, and she, at this point in her development, has 2 full-size normal X chromosomes. And on the surface of these chromosomes, we have 2 genes, and these are called X inactivation centers. Okay, in a very complicated way that we'll learn about in a chapter once we start talking about RNA. This becomes the focus of shutting down this particular X chromosome. So there's a battle going on between the 2 X chromosomes for a certain factor that we'll talk about then, and whichever one of the 2 X chromosomes randomly captures that factor, that chromosome will be turned into a bar body. So suppose the one on the left wins, then this will become inactivated and it'll become the bar body. But in a neighboring cell, maybe just by chance, the chromosome on the right captures that factor inside its cytoplasm, it will be shut down. So you can see now, if this one carries a small a, and this one carries a big A, and then this develops into the top of the arm of the baby, and this develops into the bottom of the arm of the baby, now we will have tissues that are composed of different gene products. And that is complex, but fascinating in itself. Okay, so thank you so much for listening, and we'll talk about sex-linked genes when we do the review next time.